Good morning and very welcome to this presentation of Promore Pharma's report for the third quarter of the year 2020. In a few seconds, we will hear a presentation by CEO Jonas Ekblom and CFO Erik Magnusson. Your host today, yours truly, I am Alf Riple. I work at Västra Hamnen Corporate Finance. We have the honor of hosting this as we cover Promore Pharma with our commission research service. We will first have a presentation by the company, and after that we will have a Q&A, and you will also have the opportunity to ask questions. There's a chat function in among your controls where you can write your questions, and we will read them out and the company will answer. Finally, I would also like to say that this web conference is being recorded, and the recording will be found on the company's website and on Västra Hamnen Corporate Finance YouTube channel later on. So with that, I give the word to you, Jonas.
Thank you very much, Alf. Thanks for organizing this event. I'm happy to be here. So I will give a short presentation about our company and our two main projects. So we are a small biotech located in Stockholm. We have two product development initiatives. Both of these are human peptides that are aimed for local administration. It's Sereptide, which is a phase 3 stage initiative for prevention of post-surgical adhesions after tendon repair surgery. The other program is also in principle phase 3 ready. We have very recently completed a phase 2b clinical study in this project, ropocamptide. This is a new treatment for venous leg ulcer, the most common type of chronic wounds on legs. I will start by talking a little bit about this specific program since we have completely fresh data here. On the traditional pharmaceutical markets, we're talking North America, Europe, and Japan, there are an estimated 15 to 20 million patients that have a hard-to-heal ulcer. The most common type of chronic leg ulcers on legs and feet are the so-called VLUs, venous leg ulcers. For this type of wounds, there are no prescription pharmaceuticals, and there's actually relatively few research and development programs that are ongoing. And as a reference, the average cost to treat a wound like this is somewhere between $10,000 and $15,000 US dollars. The standard treatment today is compression bandaging. The active component in our product, ropocamptide, is a human peptide that is pluripotent. It has a number of activities, a number of biological mechanisms of action. These mechanisms involve several of the different stages of the wound healing cascade. And for that reason, we think that's one of the rationale of applying this drug, because we think it can become a very effective treatment of hard-to-heal ulcers. We have very recently completed a phase 2b clinical study that has been done in Sweden and in Poland. We have randomized a total of 144 patients that have been divided into 3 arms, so a placebo arm and 2 dose strengths of LL37 or ropocamptide. This study, as most phase 2 studies, have had multiple objectives. There's been an objective to compare doses. There's been an objective to look at the efficacy of the drug, to what extent it produces a wound healing or promotes wound healing, and also, of course, to monitor safety and tolerability. The treatment time in this study has been 3 months. And for those of you who are familiar with the company know that we have done a few years back it was a phase 2a study, a smaller study, and in that study we treated the patients for 1 month, so a third of what we've done in this study. So this study is larger, and we have a larger or longer treatment duration. And I should say, after the treatment duration or completion of treatment, there's been a follow-up period of 4 months just to monitor to what extent, for instance, a wound that has been completely healed may, for instance, reoccur. When we looked at the first set of data from this study, we were a little bit set back or taken back because we actually do not see any significant, statistically significant effects of the study drug versus placebo in the main dimensions of wound healing that we have been studying. So if you look in this graph, for instance, which illustrates the frequency of patients that reaches complete wound closure after the treatment period, there is no difference between placebo and the 2 ropocamptide doses. Let me explain a little bit how to interpret these graphs. So for each treatment group, there is a dark blue column, and this column indicates the effectiveness or the outcome in the full analysis set. So those are all treated patients that contribute to data, whether that is complete or partial data. And then next to it is a green-yellow type of bar, which indicates the effectiveness or the outcome in the per protocol analysis set. So those are the patients that have completed the entire study protocol. And as you will see throughout this data set that I will show you today, these 2 bars generally go very well together, which is a good indication of good study quality. So what we can conclude from this slide is that at the global level, when we are not taking into account the different sizes of the wounds, there is no overall statistically significant effect of the treatment arms. However, the most important finding from this study came when we analyzed patients that had large versus small wounds. So in the group of patients with large wounds, and we have defined them as wounds that are 10 square centimeters or larger, and this is a common breakpoint in the literature. So there's a large number of studies that define large wounds this same way as we do. We actually see that we have a very significant improvement in the healing rate when applying ropocamptide versus placebo. So if you look, for instance, here in the left graph on the frequency of patients that reach complete closure, it goes from about 8–9% in the placebo group to close to 30% in the most optimal dose of ropocamptide. The same we can see when it comes to the number of patients that reach 70% closure of the wounds after the treatment period. And actually even more prominent is the effect when we look at the logarithmic wound healing constant for these patients. So that is a measurement of the speed of healing, where we see an effect that is close to 4 to 5 times compared to placebo of the most optimal dose of ropocamptide, and the optimal dose is statistically significant in all of these measures. So what we can conclude is we not only see a nice and significant improvement in the means when we look at these different measurements, but we also see a pattern that is very consistent with a treatment effect of ropocamptide for these type of patients, the patients with large wounds. We also, of course, monitored safety and tolerability, and what we can say in this study is that there were no severe or serious adverse events in the study that were associated with the study treatment. In the left-hand graph here, we can see the occurrence of local adverse events over time, and as you can see, there is no consistent pattern between the doses and placebo, and in fact here at the towards the end of the trial, the frequency was higher of adverse events in the placebo arm compared to the treatment arm. So there's no consistent safety concerns associated with the dose. And here on the right-hand graph, we clearly see that the vast majority of adverse events were considered to not be related to the study treatment. We've tried to reorient ourselves now when we got this somewhat surprising finding that we had strong effect in patients with large wounds. And I want to share with you some information that we have gathered over the last few days. We know from the literature that an average VLU in point prevalence can be 6-7 square centimeters approximately. We also know that the vast majority or the majority of wounds are typically small, and we also know that these small wounds generally have a better prognosis than the larger wounds. The larger wounds, and in this way again we're referring to wounds that are 10 square centimeters or larger, account for approximately 1/4 of all wounds, somewhere between 20 and 30%, depending on what population is studied. And what's interesting is that in this population, that's where the majority of the healthcare costs for these type of wounds occur. These are the patients that have wounds that have parked themselves, that take longer to treat, and the treatment per time unit is also higher in this group, driven by facts that the degree of complications, for instance, can be higher in this group. So we find it very attractive that we have a treatment efficacy in this group of wounds that are most difficult to treat, where the medical need is the largest. Before we close down or go over to the financials of the quarter, I want to share just a few words about our other project, Sereptide Pxl01. This is a peptide for prevention of post-surgical adhesions after tendon repair surgery, and we are primarily focusing on tendon injuries in the hand. Adhesions and scars can happen after essentially any type of trauma or surgery. So a new drug that can be introduced in this area, a new pharmaceutical treatment, has the potential of becoming a platform treatment. The principal mechanisms are fibroblastic adhesions and scars that essentially are produced in the same way, whether it is, let's say, in tendon repair, where a tendon attaches through permanent collagen bonds to the tendon sheet. So those are two tissue surfaces that should not be bonded together. Fibroblasts cause these adhesions. And when it comes to injuries in the hand, that restricts the movement and flexibility of the injured hand and can cause permanent loss of flexibility and mobility and strength in the injured finger. We have, since a couple of years, worked on the planning for a phase 3 clinical study for this product. We've lined up a clinical study plan for the European Union. It will be conducted under an EMA, European Medicines Agency's protocol. We will also enroll patients from India. At this point in time, we've done all major preparations for this, so we have all the major documentation more or less complete. We have completed an investigator's brochure. We have completed the clinical study protocol. We are almost complete with a production dossier. We have pre-agreements with clinics in several countries in Europe and in India, and we have a study organization with a coordinating investigator aligned. So what remains in order for us to trigger this is that we need to finance this study. Today we do not have finances for this, and that's something we are looking to address in the coming year. Shortly then, to summarize our business strategy, we have 2 projects, and Sereptide project, where we're planning to conduct a phase 3 study. We want to conduct this study in Europe and get all the way towards market authorization. That gives us the opportunity to choose to either commercialize the first indication by ourselves or through partnerships. We think that the market here is very vertical, very concentrated, so we think it's doable even for a small company as us to commercialize and sell in certain markets. So we want to keep that door open. Then when it comes to the ropocamptide project, we have recently gathered data that is positive, as I have showed you today. What we are doing now is we're preparing the final study report. We will start the preparation of a scientific publication, and we will, over the coming weeks and months, have a discussion in depth with clinical experts and experts on the therapeutic area, the business side of it, to define what is the optimal way forward for this program so that we, based on this positive data, can maximize the value for our shareholders. So with that, I would like to turn the microphone over to my colleague, Erik Magnusson, the Chief Financial Officer of Promore Pharma. Please, Erik.
Thank you. As you can see, the expenses in the quarter and also accumulated were very much in line with the costs last year. We have somewhat higher R&D costs this year compared to last year, but that is according to the clinical trial plan, so that is according to plan. The total cash flow, as you can see then, the negative result was -6.7 in the quarter versus -8.1 in the same quarter last year. The difference there is explained by that we have some passed on costs invoiced last year, and that makes the difference there. The cash flow in the third quarter was -8.6 compared to 6.8 last year, or again according to the clinical trial development. And the cash position by the end of September was 31.3 million versus the 39.9 in last of June, compared to 13 million last year. So bottom line, costs very much in line with last year, and also very much in line with our own expectations. Jonas, back to you.
Thank you, Erik. So just in a few bullet points, I want to recapitulate Promore Pharma. So we are a small company, but we have two very late-stage development initiatives, so two programs that essentially are ready for phase 3. We think that is very unique. We work with two indication areas where there today are no pharmaceutical products, so there are no prescription pharmaceuticals in these areas, so it's a large unmet medical need. We have a technology that is well validated. We have conducted several clinical studies in each of these programs, so we have retired a lot of risk that you otherwise see in early stage programs, and we have patents that bring us to the second half of the 2030s in each of these initiatives. We have a strong advantage in the fact that these products are administered locally, which means that the likelihood for severe systemic adverse events is almost completely retired. It's a very, very low probability of significant severe adverse events because of the administration mode. We work in the field of bioactive wound care, which is a field with a very high projected annual growth rate. The annual growth rate for the coming 8 years is forecasted to be 8 to 10%, which is twice of what you see in average pharma in general. And finally, we are a small company. We're just a small handful group of people that work, so we have low base costs, and we have a couple of strategic partnerships in place that allow us to operate very cost effectively. So with that, I would like to end the presentation part and turn the word back to you,
Thank you very much, Jonas and Erik. So with that, we will open up the Q&A session. You can ask your questions by writing them in the question menu that should appear among your controls on the platform. If you don't see the question menu, maybe you have to click on the among your icons. There's an arrow, a red arrow. If you click that, it will enlarge the controls, and there you will find a menu which you can enlarge and then write a question in there. We will read them out, and the company will answer. To start things off, I have a question here from Redeye analyst Fredrik Thor. He says that you mentioned the advantage of having large and financially strong shareholders. Have you talked to them recently regarding the results of the phase 2b trial, and can you say anything about their view and long-term commitment?
Erik, since you correspond to the questions with regards to finances, do you want to take the first
part of it? Sure. Yeah, we've had discussions, but we haven't really. I mean, all this report and the data here is new to us all, so we haven't really discussed on who is going to do what in the future. So that is for the future. So no comments on that at this stage.
All right.
Midrock New Technology, who is our largest investor, they have been with the company for many years now, so they are a very long-term and solid investor in shareholding in the company.
Yeah. Okay, next question. Do you still see it as feasible to get a partnership for ropocamptide? I mean, because the study didn't meet its targets, and also, was it a mistake to put positive results in the headline when it was positive? The positive was not as predefined.
There's just several ways that a question like this could be answered. It's important to keep in mind that a phase 2 clinical study has many objectives. So objectives can be to monitor safety and tolerability, its dose definition, and quite often it also has the objective of identifying ideal target segment of a drug. We did not have that as a primary objective in this study, that is correct, but nonetheless, we very clearly see when we analyze this data that an important segment responds very well to the drug. Actually, if you think about it, there are very few new medications that in any given disease area that are applicable to all comers of any disease. That is actually very unusual. But in a way, I think we have been very lucky that in a study that, even though we didn't have that as a primary objective, we already here can define a subpopulation that responds well. This is a subpopulation where I think the opportunity to price significantly a new product, it is the area where the competition is least effective. So if you would want to choose a segment, this would be the segment you would want to choose. So do we consider this as positive data? Absolutely. We've met many of the objectives that we wanted in the study. So we have identified the most effective dose. We've met study objectives in terms of safety and tolerability. And on top of that, we managed to identify a subsegment of patients where this is positive. So I absolutely think this classifies as very positive data.
While we wait for the questions, I have a question of my own, and it is knowing what you know now. If you were to design the phase 2b again, do you think you would include only patients with big wounds to begin with in the study?
Shouldn't answer firmly here, but preliminary, that would be the thought. The data we have here very clearly points to high effectiveness in the patient population with large wounds. So one opportunity, if I were to draw back the tape, so to speak, would be to make a study that would be largely focusing only on this segment. That was, of course, impossible for us to do when we started this 2 years ago, because we based the design of this study on the results we had from the prior study, which was much smaller, and it was not possible in that study to make any conclusions on the importance of the wound size for the treatment effect.
And does that also mean that looking ahead to a future phase 3, that a phase 3 study would also then be focused on the big wounds instead of a wider population?
And I shouldn't answer firmly on this because this is something we need to assess, but preliminary, that is our thought, that it might be possible to design a phase 3 very cost effectively, focusing on the target segment where we seem to have the largest effect of the treatment drug.
And also speculating further on that, if you think in terms of the perspective of the potential buyer or license taker for the drug, do you think they would be comfortable going to market with a drug that's addressing only big wounds, if that's the kind of clinical evidence that you have?
That is also an assessment that we need to take. But our preliminary opinion is that absolutely this, if anything, would make this data set even more attractive, because it suggests that one could create a very differentiated product in a segment where there actually is very little or no competition, and where the treatment costs are very high today, so there are big savings for the healthcare system. So with that comes, it gives an opportunity to actually premium price a product significantly higher than one would be able to extract from a product that would only, or would address the entire population, including the relatively uncomplicated smaller wounds. So I actually think, if anything, this would make a product line that would have a higher level of profitability than what would have been for a product addressing the entire venous leg ulcer segment. I don't know, Erik, if you have any additional comments. You've done a little bit of calculations in this regard.
I mean, yeah, sure. Just looking at the pure maths, if you, according to external sources, it's 6 to 15 times more expensive to treat a large wound compared to a small wound. And if you then anticipate that the large wounds constitute some 20 to 30% of the market, in terms of volume, the value of the large wound market would be in the range of 60 to 85–90%. So of course somebody, and that is also why we are satisfied with these results, because it actually addresses the vast majority of the value market, and I guess that it could be interest for some external partner to address this.
But at the same time, you have reported here that only 28%, or around 30, if you look at the per protocol results of even the big wounds reached full healing in the process. Is that a satisfactory result, or is that I mean, if less than a third actually are healed by the product, is that a good enough product to go to market with?
I think so. It's the 4 to 5 times higher healing speed. Many of these patients in this segment where we see efficacy have very large wounds, so not even a miracle drug can cause complete healing in 3 months. So this is, of course, in a clinical study like this, you have to define the treatment period. In this case, it was 3 months, but the average patient that was included in our study have been walking with their wounds for 5 to 6 years. So if we can cut those 5 to 6 years down to, let's say, 1 year of treatment, that would be a phenomenal saving and also quality of life implication for the afflicted patients.
All right, I have a couple of more questions from the listeners. Looking at the existing medical treatments for large wounds, what does the competition look like in the market, and what are the clinical needs?
Competition constitutes of medical device type of products. It's various bandaging products, antimicrobial ointments, and dressings. There's also negative pressure type of treatment, but there are no pharmaceutical prescription products. And particularly in the large segment where the unmet medical need is large, where many patients do not actually, with the current treatments, have any healing that is statistically significant, meaning that the wound actually opens up with the same rate as it's healing, so it's stationary, it doesn't move. So a drug like this that potentially could enhance the healing rate 4 to 5 times would cause a net decrease of the wound size, and that would be very valuable.
All right, another question. Bears say that Promore need to redo the phase 2, focusing on the subgroup big wounds to get a larger sample before going to phase 3. Why are they wrong?
Actually, I cannot say that would actually be completely wrong. We need to assess this. What we can say is that we don't feel or believe that there are any regulatory barriers to move this program into phase 3 based on these findings. However, ultimately, it's a business risk, probabilities versus risks that a sponsor would need to take, whether one would move this directly into a phase 3 setting or not. There could be a possibility of making some sort of bridging study, yes. That might save some money, but that would also lose time to the market. I'm also sure that there are various types of phase 3 designs that possibly could be utilized in the study of a group like this. Those are discussions we would need to have with regulatory authorities over coming months to have a clear picture.
I'm thinking also that a successful study here in Sereptide was part of your plan. You planned to leverage on that to raise financing for going into phase 3 with Sereptide. Now that it might take longer before you have that firm result, or maybe a licensing deal for Sereptide may take longer. What are you thinking about the timing for when you can start phase 3 in Sereptide?
Right. So, Erik, if you can take the financials, I can just comment in general that we feel that these results are really good. We need to understand how we package this to make it appetizing for a potential license taker. We want to keep all doors open because, of course, we're interested in divesting a license for this program that would potentially generate milestone payments and license fees. But on the other hand, with good results like this, we don't want to sell it at any price, so to speak, because that would be giving away important value or leaving money on the table. So we don't want to do that. For that reason, we want to evaluate all possible strategic avenues that we may have. In respect to the financials, perhaps you can say a few words, Erik.
Yeah, I mean, we've already touched upon this. This is quite new information to us as well. So we have just started to evaluate all the kind of possibilities that we have. So we will simply have to get back a little bit later on exactly how we would like to address this in the future.
All right. Another question.
Expectation, right, with the market, it's unlikely that we within weeks or a few months will secure a deal. That typically takes some time. We plan to make a final study report on this. We plan to make a peer-reviewed scientific publication of it, and typically contacts with larger companies that might be interested will go in several steps. So there is a deal cycle time to take into account. I just want to avoid any misunderstanding. It's not that we will have a deal happening by the end of the year here on this. These are fantastic data, but it will take a little bit of time to reorient ourselves, package it, and find the appropriate license taker for projects like this.
I don't want to drill repeatedly into a question that you maybe can't answer, but it would be interesting to hear you say what I mean, if you think that this will affect the timing of phase 3 and Sereptide in any way.
So I understand your question. Are you asking whether the outcome of the LO37 trial will have an impact on the start of the Sereptide phase 3 program? Was that your question?
Exactly. I mean, considering that I assume it will take longer to license out Sereptide given these results compared to if you had a home run of significance in the whole population. So, I mean, given that it will take longer with these results, do you think that will cause a delay in starting phase 3 in Sereptide?
I'll answer the question indirectly. We will have roughly 21 million Swedish crowns on the bank by the end of this year. That is not sufficient to operationalize the phase 3 program for Sereptide. So we will, during next year, need to have a capital increase in the company in one way or another in order to start the program. We will naturally do everything we can to minimize that we have any lag time or delayed time. We will work with maximum speed and able to operationalize the Pxl01 in Europe.
Okay, thanks. A couple more questions. Is the dosage for Sereptide of 5? It should be 0.5 milligrams per milliliter. Is that correct? Is that fixed?
If I said 5, then I said wrong.
All right. So is that fixed now? Are you tempted to look at small variations, say 7 or 8 milligrams, for instance, or 0.7, 0.8?
No, we feel quite confident that this is an optimal target dose based on what we have done. We think it would be futile. It would be no meaning in making smaller dose increments, because the power of statistics that would be required to show differences between 2 doses like that would be require a very large number of patients. So we think that with this study, we have actually successfully identified a main concentration to work on for phase 3.
Great. I haven't received any more questions from listeners, and I have reached the end of my questions. So I don't know if there's anything you would like to add, Jonas or Erik, before we close the meeting.
No, I don't think so. It's been a very interesting session, very good questions, and thank you for organizing this off.
You're very welcome. So we will probably talk again in 3 months' time.
Sounds very good.
Thank you. Bye-bye.
Bye-bye.